Hey chromosome fans, it's me, Dr. Malone, here to talk about translocations. And here we go. Sorry, I don't have anybody interesting in my office right now, so I apologize if this is boring. But hey, you get what you get. Alright, a translocation, alluded to earlier, when one piece of one chromosome attaches to another. Two main types that we care about, or that are medically relevant: reciprocal and Robertsonian. One being balanced, an exact switch from chromosome to chromosome, and the other one not. Both can cause big fat problems, and we're going to talk about why. So here we go. We're going to look at non-reciprocal, which means they don't switch back and forth, and reciprocal. These are the ones that are the usual translocations we talk about. Interchromosomal, which means between two different chromosomes. Non-homologous. What is the switch of a part, of one chromosome to another chromosome when they're homologous chromosomes? Anyone? Anyone? Crossing over! Right? That's the non-sisters, the switching parts. That's supposed to happen. Switching parts between non-homologous chromosomes, like chromosome 14 and chromosome 2, not supposed to happen. Causes problems. So that's what it looks like. We've switched this guy onto here, and this guy onto here, and that equals that. Some other terms that I don't necessarily care about, but you should know. Non-reciprocal intra always means within. Within. Intra. No matter what in the English language, that's what it means. Inter always means between. Okay? Okay. Intra, within, inter, between. And that's just one piece jumps from one onto another, right? He jumped onto there, and that's what he looks like, and that's what he looks like. Or this guy just moves down here, and now he looks like that. Okay? Good. Yay. That's easy. A Robertsonian translocation is specifically a type of translocation that tries to be reciprocal, but ends up not being balanced because one entire chromosome is lost. Which means this has to happen between a short arm of an acrocentric chromosome. What does that mean? Do you remember that term? Just means the centromere is really close to one end. And is exchanged with the long arm of another. So it has to be two different chromosomes that both are acrocentric. So two big pieces, right, this side and this side come together, and this side and this side come together. So now this is a whopper, has almost the entire chromosome from each, and the little pieces come together, and it's usually lost. It fails to segregate, is lost, it generally has trouble attaching to its spindle fibers. Okay, so that would be during meiosis. And that would be very bad, because then we are going to have an aneuploidy. Yes? Yes. Okay, so this figure just shows us a regular reciprocal translocation, not Robertsonian, so this is not Robert. It's just recip. Okay? And all it's saying is that when we try to do meiosis, when we pair up, remember we talked about the pairing of homologous chromosomes has to do with complementary base pairing and what genes are similar, and that's how the chromosomes find each other, because they have the same genes on them in the same order. Now that they have chunks of one on another, right? This red is part of the blue and the blue is part of the red. When they line up, they still use the complementary base pairing in the genes to align, and then that isn't what it's supposed to look like at metaphase. And so then sometimes it gets mixed up on what spindle fibers it grabs. And when we go through anaphase 2 to pull the sisters apart, a lot of times it gets screwed up. I will not ask you to show this. No applied here. So I will not give you an example and ask you what the gametes will look like. I just want you to understand that they're screwed up and translocations are bad. Totally messes stuff up. Robertsonian translocations. Let's talk about that because it does cause a form of Down syndrome that's actually inherited. Normally Down syndrome is spontaneous, but in this case it is inherited. Much of chromosome 21 is attached to chromosome 14. And so it's really that third copy of 21, even though it's not the entire chromosome, that causes Down syndrome. So if we have a normal parent right here in the P generation with normal 21 and normal 14, and we have a carrier parent who has one normal 21, one 14-21 translocation, and a number 14. This person looks normal. If you count their chromosomes, they have two 21s, missing a little bit, and two 14s, one missing a little bit. But when this individual makes gametes, it doesn't come out exactly right. There aren't two 21s and two 14s. And so our gametes can look like these, depending on, right, all the different possible ways they can segregate. Remember, independent assortment. This guy's gamete, of course, is normal. And then the zygotes, if this fertilizes this guy, you get a translocation carrier. If he fertilizes this guy, looks totally normal. This guy, Down syndrome. Because look, there are 3 chromosome 21s, the blue ones. 1, 2, and most of that one's 3. This normal guy with that, it's a monosomy. There are never any monosomies aside from sex chromosomes that ever survive, so this is lethal. This normal guy over here with this one, again, trisomy 14, not ever survives, and this one with this one, monosomy 14, again, lethal. So this type of translocation can be completely normal in a carrier, but again, this person as a parent has a probability of having a Down syndrome child. I do want you to be able to apply the Robertsonian. You should know this slide. Know this slide. Because you may be asked to draw gametes. What happens if they're fertilized? Does it have Down syndrome or not? What are all the possibilities? If you see a zygote, could you tell me if it's trisomy 21? Yes? Yes. Hello, my friends. This is an example of a reciprocal or balanced translocation that causes a disaster. This is a follicular lymphoma. It's cancer. It's caused by a translocation between human chromosome 14 and 18. In this case, we're only showing one of the chromosomes, the one that causes the problem. The same thing happens. The translocation is balanced. They switch places. We're just not looking at the other chromosome because that one, as far as we know, doesn't seem to cause a big problem. This one does, however. So it's a specific break at a specific point in a gene that has an enhancer. We will talk about enhancers when we get to gene regulation, but quickly it turns up transcription like craziness, way, way, way up. So gene expression explodes when you have an enhancer. And so chromosome 14 is This original gene here makes antibodies. Antibodies fight disease. Antibodies are needed when you have a bacterial infection, and we need a crap load of them all at once. So this gene has an enhancer. When it gets the signal to go, you just make a buttload of antibodies. So in this translocation, this enhancer is moved into a chromosome, chromosome 18, in front of the BCL2 gene, which essentially makes a shit ton of BCL2. BCL2 causes a cell cycle to go, and poo, we get cancer in B cells where this is expressed. All bad. Another bad example of reciprocal or balanced, again causing cancer, is CML. CML used to be a death sentence. There's now a really great drug for it, but that's beside the point. Again, two different genes, essentially this time within each one in the coding region. These two switch places. Again, we're only looking at one of the chromosomes. The other one, the same thing happened. It's completely balanced, but that one doesn't seem to cause a problem. This creates a fusion gene, fusion protein. It cut and pasted in frame. How the hell did that happen? So when that happens, we make a protein that doesn't exist in any other type of cell, and it does cause the cell cycle to go. The nice thing, I mean, if you could call any kind of translocation or cancer nice, is that this protein itself does not exist anyplace else in the body except in these translocated cells, because it's this crazy fusion protein. Now the ABL gene exists and the BCR gene exists, but not this hybrid. So if we target a drug to the region that doesn't exist anyplace else in our bodies, that the drug recognizes this and then attacks these cells and kills them, it's a cancer treatment that doesn't affect any other cells, no side effects, none of the crazy chemo hair falling out and getting sick and all that stuff. So there is a drug, and it does work, and now CML is highly curable, quote unquote, can put people into remission essentially forever. So from a complete disaster to something great, all based on knowing the science and scientific research. Science saves lives, people. All right, so here's a little screenshot to upload. This one, I believe, if I look it up, is called translocation. It's called iPad, I don't know, 12 point something or other. iPad 12.1, translocation. So do this, upload, and get some points. And I'll see you soon. Thanks for listening. Bye-bye.